Hi everyone. In the CPGET Microbiology Important MCQs, in this video, we are going to discuss the Series 20, which poses 25 important questions. Let's begin. First question. A unicellular blue-green algae that fixes atmospheric nitrogen is, options are A. Oscillatoria, B. Anabina, C. Nostoc, D. Cleotheca. And the right answer here it is going to be the Anabina, as this Anabina is a blue-green algae that is nothing but the cyanobacteria, and it is going to be a unicellular prokaryotic organism as it do not possess any cell membrane bound organelles or a true nucleus. So that's the reason why the cyanobacteria is going to be considered as a prokaryote, and this Anabina is going to be used as a nitrogen fixing bacteria which converts the nitrogen into ammonia and make it available to the plants. Second question. Anaerobic breakdown of proteins by microorganisms is known as, options are A. Putrefaction, B. Curing, C. Retting, D. Pasteurization. And the right answer here it is going to be the putrefaction, where the putrefaction is going to be referred as an anaerobic decomposition of organic waste, particularly proteins that lead to the formation of foul smelling because of incompletely oxidized products. Third question. The synthesis of polypeptide chain according to the codons present on mRNA is known as. So simply the synthesis of polypeptide chain, obviously we call it as translation. So options given are transduction, translation, transcription and transformation. The transduction and transformation are related to the gene transfer mechanism, whereas the transcription is a synthesis of mRNA. So the synthesis of polypeptide chain or protein synthesis is nothing but the translation. Fourth question. Leaf blight of rice is caused by, options are A. Pyraclaria oryza, B. Heminthosporium oryzae, C. Streptomyces oryzae, D. Xanthomonas oryzae. And the right answer here it is going to the Pyraclaria oryzae. So coming to the other type of the diseases, the bacterial blight is caused by Xanthomonas oryzae, and it is going to cause the wilting of seedlings and yellowing and drying of leaves. Then the blast disease is going to be caused by the Pyraclaria oryza, that is nothing but the leaf blight disease. Then the Coccobolus miabenus is formerly called as a Heminthosporium oryzae, is a fungus that causes brown spot disease in rice. Fifth question. Microorganisms involved in the production of yogurt. Options are A. Leuconosta cremoris, B. Lactococcus lactis, C. Streptococcus thermophilus, D. Lactobacillus acidophilus. And the microorganisms involved in the production of yogurt is going to be the Streptococcus thermophilus as well as the Lactococcus bulgaricus. So these are the two species in the ratio 1:1 we are using in the production of yogurt. Sixth question. Complications of the typhoid fever are, options A. Intestinal perforation, B. Meningitis, C. Bone abscesses, D. Neurological damage. So the typhoid fever is a type of enteric fever which causes the intestinal damage. So obviously it is going to lead to the intestinal perforation. Seventh question. The rancidity of stored food is due to the activity of, options are A. Lipolytic microbes, B. Proteolytic microbes, C. Toxigenic microbes, D. Saccharolytic microbes. And the right answer here it is going to be the lipolytic microorganisms. That means the rancidity of the stored foods is due to the activity of lipolytic microorganisms. Eighth question. A non-legume plant that forms nitrogen-fixing nodules with rhizobium. Options are A. Groundnut, B. Oryza sativa, C. Parasponia, D. Maize. So out of all these things, the one which is a non-legume plant is a Parasponia. So this is the one which is going to form the nodules with rhizobium. So Parasiponia is the only non-legume host plant known to be nodulated by rhizobia, and the host plants are medium-sized tropical trees and pointed species growing on nitrogen-poor and disturbed soils. And these trees originated from the Malaya archipelago. Ninth question. A fungus that is employed as biopesticide is. Options are Trichoderma viride, B. Bacillus denitrificans, C. Bacillus thuringiensis, D. Nuclear polyhedrosis. So they are asking about a fungus. So here the Trichoderma viride is the one which is going to be used as a bioinsecticide or biopesticide in the biological control of insects and pests. So Trichoderma viridans is a fungus and a biofungicide, which is going to be a biological control agent, have been widely used against many plant pathogens such as virus, bacteria, fungi, nematodes, and higher parasitic plants. Then coming to the nuclear polyhedrosis, which we call it as NPV. This virus is belonging to the family called as baculovirus, and it is having a capacity of affecting the insects, predominantly moths and butterflies. So it can also be used as a pesticide. Then we know the BT, that is Bacillus thuringiensis, which is the derivative of bacteria, which is going to produce some sort of toxins, which is going to kill the mainly the insect orders or the nematodes. Then Thiobacillus denitrificans is the one able to reduce not only the nitrate but nitrite as well. So this Thiobacillus, there is no Bacillus thuringiensis, it is a Thiobacillus denitrificans, also being considered for sulfide removal in sulfide rich waste sources such as in sour gases and waters and in spent sulfidic causetics. 10th question. Which of the following molecule contains Shine-Dalgarno sequence? Options are A. mRNA, B. tRNA, C. rRNA, D. DNA. So we know that Shine-Dalgarno sequence is going to be present in the mRNA molecule. So this prokaryotic mRNA molecule, as we know, it is a polycystonic and contains this sequence that is going to be called as Shine-Dalgarno sequence for ribosome binding. And in the eukaryotic mRNAs, as they are more complex and generally contain a cap and a poly A tail. 11th, preservation of bacteria by vacuum drying is known as. Options are A. Evaporation, B. Lyophilization, C. Etching, D. Liquid nitrogen storage. So here they are asking about the preservation. So evaporation means just removing the water, so that is not going to be combined with the vacuum, so it is wrong. Then etching is also another type of the thing, and liquid nitrogen storage is directly putting the vials in the liquid nitrogen. So the one and only preservation of bacteria by vacuum drying method is lyophilization. So here once again have a glance of the things that I told you. Freeze drying, also known as lyophilization or cryodesiccation, is a low temperature dehydration process, and then that is going to involve the product lowering the pressure, then removing the ice by sublimation, and this in contrast dehydration by most conventional methods that evaporate using heat. Then etching is traditionally the process of using strong acid or mordant to cut into unprotected parts of a metal surface to create a design inside in the metal. Then cryopreservation of bacteria at temperatures between -90, that is 196 degrees, and -150 in or above liquid nitrogen is a reliable method for long-term storage. So here glass capillaries can be stored in the gas phase of liquid nitrogen as well as in the liquid phase. 12. Bacteria are included in the kingdom. Options are A. Protista, B. Mycota, C. Plantae, D. Monera. And the right answer here is going to be the Monera. And we know that Monera is a kingdom that contains unicellular organisms with a prokaryotic cell organization that is having no nuclear membrane such as bacteria. So obviously our bacteria is included in the kingdom Monera as they are single-celled organisms with no true nuclear membrane. 13th, the virus that replicates through lysogenic cycle. Options are A. T4 phase, B. HIV, C. Lambda phase, D. rhabdo phase. So out of all these things, the lambda phase is the one that is going to replicate by lysogenic cycle. Yes, this is also going to have the lytic cycle also, but majority of the temperature phases such as lambda phase can reproduce using both lytic and lysogenic. But via the lysogenic cycle, the bacterial phage's genome is not expressed and is integrated into the bacterial genome to form the prophase. So that means the combined form of the bacteria DNA and the viral genome is going to be called as prophase stage. 14th, base pairing in DNA molecule is. Options are A. Adenine uracil base pair, cytosine thymine base pair, adenine guanine base pair, and adenine thymine base pair. And we know that the DNA do not possess the uracil, so the option A is wrong. Then you have to remember that guanine always pairs with the cytosine. So here the cytosine thymine base pairing is also wrong. Then adenine always pairs with the thymine, so here they have given the G, so option C is also the wrong answer, and the right answer here it is going to be the adenine thymine base pairing. 15th question, electron microscope was invented by. Options are Robert Hooke, B. Jernik, C. Leeuwenhoek, D. Nolan Ruska, and the right answer here it is going to be obviously the Nolan Ruska. So the invention of the electron microscope by Max Noll and Ernest Ruska at Berlin in 1931 finally overcame the barrier to higher resolution that had been imposed by the limitations of the visible light. In 1953, Jernik won the Nobel Prize in Physics for the invention of which microscope? Phase contrast microscope. Noll and Ruska are the electron microscope invention, whereas Jernik is going to be of invention of the phase contrast microscope in the year 1953, with which we can study the internal structure without the need of staining and thus kill the cells. So then moving to the Robert Hooke, we know very well he is the person who discovered the cell structure of a cord, that is a cell. And not only that, he also involved in the study of microscopic fossils, and he published a book which is very known to you, that is Micrographia, in which he includes the sketches of various natural things such as what we call as the structures of these dead cells, all these things by whatever the things that he observed under the microscope. Then coming to the Leeuwenhoek, we know that he is the first observer of microscopes, microorganism, through his special made single lens microscope, and those creatures, the small minute creatures, he called them as animalcules. 16th, the term or the time required at a specific temperature to kill a population of cells is expressed as. Options are A. D value, B. Z value, C. F value, and D. P value. So here this question is a little bit tricky, and there may be a possibility of asking this question in many exams, because the time required at a specific temperature to kill a population of cells or spores is expressed as. The right answer is going to be the D value. That means the D value is the time required to kill a population of cells, that is nothing but the microorganisms, in or at a given specific temperature in a specific medium. Then we call it as D value. That means the D value is going to give the time needed to kill an organism at a given temperature. Then what is the Z value? The Z value is going to be the increase or decrease in temperature required to reduce or increase the decimal reduction time by 1 decimal. Simply, it relates the resistance of an organism to differing temperatures. So at which temperature it is going to be of resistant, at which temperature it is going to be of sensitive, that relation is going to be given by the Z value. Then what is meant by F value? F value is going to be a process in which the number of the microorganism of minutes required to kill a known population of microorganisms in a given food under specified conditions is going to be called as F value. So instead of the time required, if they ask you the minutes required at a specific temperature to kill a population of the cells, known population of the cell, then you have to give the option as a F value. But here in general they have asked you the time required, so obviously it goes with the D value, right answer. So here you can see what is meant by p-value. The p-value is a measure of probability that observed difference could be occurred just by random chances. So these are the terminologies that you have to remember, because sometimes they may ask you about the Z value, sometimes they may ask you about the F value also. 17th, red rot of sugarcane is caused by. Options are A. Fusarium, B. Xanthomonas, C. Erwinia, and D. Collatotrichum. And the right answer here, it is going to be the Collatotrichum, where you are going to have the Collatotrichum falcatum vent is the one which is responsible for causing this red rot of sugarcane. Then coming to the fusarium, this fusarium is going to cause a fusarium wilt in crops such as sweet potato, legumes, melons, and banana. And Xanthomonas is going to, for example, the Xanthomonas citri is able to cause the citrus canker in the citrus plants, and majority of these Xanthomonas are going to cause a disease in plants such as tomato, pepper, cabbage, banana, and bean. Then Erwinia. So we know that Erwinia is going to cause a soft rot in plant. The best example of the species is Erwinia catovora. 18th question. Disappearance of blue color by taking more time in MBRT of milk indicates. Options are A. Good quality of milk, B. Poor quality of milk, C. MBRT not related to the milk quality, D. None. So read the question properly. Disappearance of blue color by taking more time, they are asking. So as soon as you see the question, you think that is this going to indicate the poor quality. No, they're asking taking more time. So if the disappearance of the blue color, that is methylene blue color, is going to take more time in the methylene blue reduction test of milk, is going to indicate the good quality of the milk. As the decolorization is going to take more time, that much of the quality of the milk is going to be ensured. 19th question. Wobble hypothesis was proposed by. Options are A. Crick, B. Watson, C. Corana, D. Baltimore. And the right answer here is going to be the Crick, who proposed this Wobble hypothesis. So the Wobble hypothesis is going to explain why multiple codons can code for a single amino acid. That means one tRNA molecule can recognize and bind to more than one codon due to the less precise base pairs that can arise between the third base of the codon and the base at the first position of the anticodon. And then coming to Mr. Watson, we know that together Watson and Crick discovered the DNA structure, that is a double helix structure. Then Corana Harbogobin Corana is a biochemist who won the Nobel Prize because of the construction of first synthetic gene, and he also renewed research in the nucleic acids and proteins. Then coming to the Baltimore classification is well known to us of viruses, where he classified the viruses into 7 groups depending upon a combination of their nucleic acids, strands, sense, and method of replication. Then moving to the 20th, in the soil environment, most bacteria are found in. Options are A. the deepest part of the soil, B. within first few millimeters, C. only sand is present, D. water availability in foods. So here you have to remember the soil environment they are asking. So only sand is wrong, water availability in foods is wrong, and the deepest part of the soil is also not going to possess. So within the first few millimeters of the soil, they are going to be of having more number of the bacteria because of having the more humus in that region. So obviously where the dead and decaying composing matter will be there may be the chances of having the more number of bacteria. So in the soil environment, most bacteria are found in the first few millimeters. 21st, Vibrio cholera is. Options are A. gram-positive bacilli, B. gram-positive cocci, C. gram-negative cocci, D. gram-negative bacilli. And here the Vibrio cholera is a gram-negative bacilli. And it is a short curved motile, so we will call it as a comma shape, that produces enterotoxin, a protein that induces hypersecretion of an isotonic electrolyte solution by the small bowel mucosa. And humans are the only known natural host of Vibrio cholera. 22. Diseases that are constantly present in a population are epidemic, endemic, pandemic, and sporadic. And the right answer here, it is going to be the endemic. Diseases that are constantly present in a population are endemic. So what about the other terminologies? The sporadic is going to refer to a disease that occurs infrequently and irregularly. They are present constantly in the population but infrequently and irregularly, then we call it as sporadic. Then endemic means constant presence, usually prevalence of a disease or infection agent in a population within a geographic area. Then hyperendemic refers to persistent high levels of the disease occurrence, or diseases that are constantly present in a population within a particular geographic region are called endemic diseases. Then what is meant by epidemic? Is the disease that if the diseases are going to affect a large number of people with a community, population or region, then we call it as epidemic. Then what is a pandemic? So a pandemic is an epidemic, that means it is going to affect the large number of the people that spreads over multiple countries or continents. The best example that is going to be the coronavirus or corona disease is going to infection is the one example of pandemic where it is going to be of affecting the large number of people throughout the countries, throughout the continents. That's the reason why we call the coronavirus infection as pandemic. 23rd, example of antiviral antibiotics. Options are A. erythromycin, B. penicillin, C. chloramphenicol, D. amantadine. So here to confuse you, they have given the antiviral antibiotics. We know that the erythromycin, penicillin and the chloramphenicol are a type of antibiotics which will attack only the bacteria and the other parasites except the viruses. But here the antiviral agent is going to be called as amantadine. So this amantadine is an antiviral medicine used to prevent certain influenza, that is flu infections of a type B. And it may be given alone or together with flu shots. And amantadine will not work for colds, other types of flu or other virus infections. Whereas erythromycin, we know this is a group of medicines called macrolide antibiotics, and these macrolide antibiotics work by killing the bacteria causing the infection. And this erythromycin is mainly used to treat the diseases caused by the bacteria like bronchitis, pneumonia, legionnaires disease, that means a type of infection, and pertussis. Then penicillin, we know it is a narrow range antibiotic that covers gram-positive bacteria and some gram-negative bacteria. Then chloramphenicol is a broad-spectrum antibiotic derived from the Streptomyces venezuelae with primarily bacteriostatic activity. And this antibiotic is useful for the treatment of diseases such as conjunctivitis, that is eye infection, or by mouth or by injection into the vein, used to treat the meningitis, plague, cholera and the typhoid fever. See, I'm giving the explanation regarding the other antibiotics also. For example, in the examination, they may ask you chloramphenicol is a dash type of antibiotic, whether it is a narrow spectrum, broad spectrum, like that. So then you have to remember that it is a broad spectrum antibiotic. For that reason, I'm giving the explanation to the other options also. 24. The hexose monophosphate shunt and enterodeodorov pathway. Options are A. DNA synthesis in microorganism, B. photosynthesis in photosynthetic organism, C. energy metabolism in certain species of microorganism, D. chemiosmosis. They are asking what is the use of HMP pathway and ED pathway, that is enter and deodorized pathway. So obviously these are going to be the alternative pathways of the EMP pathway, that is a glycolysis, and these are the type of glycolysis where the glucose is break down to release the energy. So obviously the right answer here, it is going to be the energy metabolism in certain species of microorganisms. And the right here, HMP is also called as a PP shunt or pentose phosphate shunt because the pathway allows for carbon atoms from glucose 6-phosphate to take a brief detour, that means a stunt, before they proceed down the EMP pathway. Then the enter-deodorized pathway are going to occur only in the prokaryotes, so that is a specialty, and it uses 6-phosphogluconate dehydratase and 2-keto-3-deoxy-phosphogluconate aldolose, so that we call it as KDPG, to create pyruvate from glucose. By comparison, glycolysis has a net yield of 2 ATP and 2 NADPH for every 1 glucose molecule processed. 25th, a bacterial arrangement in packets of 8 cells is described as. Options are A. Micrococcus, B. Diplococcus, C. Tetrads, D. Sarcina. And the right answer here, it is going to be the Sarcina, where the bacterial arrangement in packets of 8 cells is going to be described as a Sarcina, whereas a 4 is going to be treated as a Tetrads. And the micrococcus are also going to be of a tetrads or irregular clusters, but not in chains. Okay, and diplococcus, obviously we know the diplococcus are going to be 2 or in pairs. So this is all about the CPGET 20 series. Thank you.